Selection of honeybees, aiming for vitality. Bees pollinate many flowering plants, thereby aiding environmental diversity and the economic success of crop farming. Today, beekeeping is possible even in densely populated areas, thanks to the systematic selection for gentleness. Due to the occurrence of new illnesses, selection for vigorous and healthy colonies represents an exceptional challenge for breeders. Amongst the illnesses and parasites of the honeybee, the varroa mite above all causes a substantial burden. Varroa mites afflict not only adult bees, but also the brood. A high infestation produces characteristic damage. The brood surface isn't solid. One finds opened cells and dead pupae. Occasionally one finds varroa mites still there. They infiltrate the cells a few hours prior to capping and reproduce on the adolescent bees. A sure sign of parasitizing mites are the white fecal piles on the top cell wall. Sometimes bees emerge from the afflicted brood cells with shortened abdomens and crippled wings. This disfigurement results from the withdrawal of nutrients and the transmission of viruses from mites. During the brood phase of the bees, the varroa mites multiply. The infestation of a colony can double monthly. In many cases, during summer, treatments must be applied to ensure the survival of the colony. However, in some colonies, the infestation grows much more slowly. To evaluate its development, the infestation is determined at the season's beginning and at the start of July. Colonies with minimal varroa increases are preferentially scored for selection. In spring, pussy willows are the first abundant pollen source in many locations. The bees can now gather large amounts of pollen and begin to rear brood. With the start of the brood season, the reproduction of mites also commences in the colonies, so this is the appropriate time to evaluate the starting infestation. For this purpose, at the start of the pussy willow bloom, all colonies of this test apiary received a mite drop tray, which should cover the entire bottom board area. The tray must stay in the colony for at least 3 weeks and is monitored weekly during that time. On the light base, the varroa mites are recognized effortlessly and can easily be counted. Date of control and mite count are noted on the record form. In the first 10 days of July, the varroa infestation is evaluated again. Meanwhile, it has increased so much that it can be gauged from a single bee sample. The parasitization of bees in the honey supers is more uniform than in the brood boxes. So the sample for the analysis is taken from here. There's also no danger of damaging the queen. As the bee sample evaluates the relative infestation, this method is suitable to compare hives of different strengths. First, the bees are shaken onto a sheet of plastic. For reliable results, the sample must contain at least 30 grams of bees. By folding the sheet, the bees can easily be poured into the specimen cup. The cup is marked with the date and colony number. Before evaluation, the bees are killed in the freezer. After determining the weight of the empty specimen cup, the weight of the sample is calculated. A digital scale that accurately measures to 1/10 of a gram is ideal. In soapy water, the mites detach from the bees. First, the sample is transferred to a larger container. It's important not to leave any mites behind in the specimen cup. Water and some detergent are added. Then a brief agitation ensures that all bees are wetted. After at least 15 minutes, all of the mites have detached themselves from the bees. The sample is shaken vigorously once more and emptied into a double strainer. Again, it's important not to leave any mites in the container. The bees in the strainer are sprayed with a forceful jet of water. The coarse strainer holds back the bees while the mites fall into the fine strainer. From there, they're shaken onto a light-coloured pad. There they can be counted quickly and reliably. The results are recorded along with the weight of the bee sample. The infestation development depends on a variety of factors. Parasitized bees often leave their colony and thus help to minimize the varroa load. Grooming is another protective reaction. Bees find attached mites irritating. They react with forceful shaking and try to cast off the parasites. Mites only reproduce in capped brood. In the infestation development, brood size and brood cycle of a colony play a significant role. Brood cells also vary in their attractiveness to varroa females. An additional mechanism of defense exists in the opening and cleaning out of afflicted brood by nurse bees. The parasitization leads to disturbances and changes in the brood cells. But currently, only little is known about the actual signals to the nurse bees. Through the participation of several bees, the afflicted cell is cleaned out. Even if the mite can often escape, she is disturbed in her reproduction. The needle test is a quick and simple method to evaluate brood hygiene. It must be executed at least twice. Within a marked region, 50 capped cells are punctured with a fine insect pin. It's important to puncture them all the way to the cell bottom. After 8 to 12 hours, the colonies with pronounced hygienic behaviour will have opened most of the cells and removed the brood. The percentage of completely cleaned out cells serves as a comparison of the brood hygiene of individual colonies. At the end of the testing, all data are noted in the breeding records. In In addition to all of the performance data, the start and finishing mite infestation and the average of all completed needle tests are included in the testing report. On the basis of these data, breeding values can be calculated for individual traits. During the performance test, colonies demonstrate their varying characteristic traits. Most of the attributes are distributed normally, that is, few individual colonies have an especially poor or good trait. Many, however, are average. The term selection means taking the best pick of the animals for breeding purposes. The selection difference determines the superiority against the average and depends on the distribution of the trait as well as the selection intensity. Heredity determines to what extent the selection difference is passed on as stock improvement to the next generation. Through progressive selection, the disposition of the animals becomes ever more unified. The still possible stock improvement decreases from generation to generation. The time frame over which one can select effectively and the magnitude of stock improvement depend substantially on the size of the population. A single operation with limited breeding colonies reaches its limits after 3 to 5 generations. Fundamental improvements of the genetic makeup, such as Varroa tolerance, thus require selection in large populations of more than 1,000 colonies. If from these approximately 100 are used annually as breeding colonies, a continuous stock improvement is possible over decades. To achieve such a large population, many breeding operations must cooperate. In the tolerance breeding work group, they work according to uniform criteria, exchange test queens, and evaluate all data via a central breeding estimation system. The best colonies out of the performance tests qualify for the vitality test. This test is preferably carried out with the cooperation of the bee institutes at central testing stations. The delivery takes place at the end of July, early August. Selection for Varroa tolerance aims for colonies that can survive on their own. That's why colonies chosen for the vitality test are set up and observed in an isolated apiary without treatment. In addition, further tolerance parameters are examined, such as the brood infestation and the susceptibility to virus infections. After the flight entrances are opened, the colonies can accustom themselves to the new location for a few days. To guarantee a uniform handling and evaluation during the vitality test, all colonies are transferred into identical hive bodies after 2 to 3 days. This simplifies additional work such as regular controls and feeding. To preserve the established nest structure, all combs are placed according to their former order. During comb transfer, the beekeeper gets a close impression of the colony condition at delivery. Starting in late August, the apiaries are checked in a weekly rotation. Weak colonies can thus be recognized in time and broken down. Thus, robbing and the resulting spread of pathogens can be avoided. First, one estimates colony strength and counts the occupied frames. In this case, there are 15. When feeding hives for winter in early to mid-October, the colonies should occupy at least 8 to 10 frames. For a more accurate evaluation of the Varroa infestation, a brood sample is removed. Only older capped brood is suitable. The sample should contain approximately 400 cells. During later examination in the lab, the proportion of reproducing mites and the number of infested brood cells are determined. In late August, the brood infestation should not exceed 5 to 8%. A higher infestation could damage the winter bees and weaken the overwintering ability of the hive. In coordination with the brood rhythm of the hive, removal of samples takes place every 3 weeks. From a brood-free frame, a bee sample of at least 30 grams is removed. From the brood and bee samples, the total infestation can be calculated. In addition, the bees are examined for virus infections. Together with the Varroa mite, the acute bee paralysis and deformed wing viruses influence the survivability of the colonies. Lastly, controlled date and colony strength are recorded. In late March, early April, the colonies are again monitored after overwintering. Some have not survived the winter, others have become much weaker.